Hi everyone, welcome to my channel Jithender's Biology. Today's topic, Unit 2, Cell, the basic unit of life. Unit 2, Cell, the basic unit of life. You know that this world is made up of with various types of organisms. In them, very minute organisms like what? That is mosses. Whereas there will be also very large organisms like blue whales. So we have very minute organisms like mosses and very large organisms like blue whales. But wherever it may be a minute organism or it may be a large organisms, all are made up of with the cell. So that's why we can say the basic unit of life. The basic unit of life is what? Cell. Why? Because it may be a tiny organisms or it may be very huge organisms. They have been made with the cell. So that's why we can say cell is the basic unit of life. And before 350 years ago, there was no microscopes. Okay, there was no microscope. As there is no microscope, we can't see the invisible world. The invisible world or invisible organisms like microbes. Invisible organisms like what? Bacteria, fungi, etc. Protozoans, etc. which are not visible to our naked eye. We are unable to see. Why? Because they are able to see with only help of a microscope. So that's why the invisible world of organisms like bacteria, fungi, protozoans, okay, that all we are not able to see. But after the discovery of microscope, we have been known about still several minute organisms like bacteria, fungi and etc. And moreover, Nana, these organisms are like this we seen. And moreover, if you know about the cell means there are some scientists who has been contributed a lot for the research of the cell. Who are the scientists? The scientists like Creature, Swammerdam, as well as that is Leuwenhoek, Antony van Leeuwenhoek, as well as one more person, that is a Robert Hooke. So these are the, of course, still there are so many number of scientists who has been contributed a lot, etc. But in them the worthy notified scientists who has been said some information about the cell are these. Who are they? Athanas Creature, Jan Swammerdam, Leuwenhoek, Antony van Leeuwenhoek, as well as a Robert Hooke. And among these, first we have to discuss about Antony van Leeuwenhoek. Antony van Leeuwenhoek. Antony van Leeuwenhoek. Actually, this Antony van Leeuwenhoek is a scientist who belongs to Netherlands, and as he was a leather merchant who actually do the leather business. And so this Antony, but he was basically interested in observing of that is microorganisms, and he has been developed in his with his own that is magnifying lenses. He has been developed what on magnifying lenses which is having 300 times of magnification. 300 times of what on magnification power. And with the help of this magnifying lenses, actually magnifying lenses means what? And a lens which will increase the size of an organism than its original size is called as a magnifying. Magnifying means increasing this from its original size. Like that, he was the first person to observe with the help of his magnifying lens. The first person to observe what is that? Bacteria, as well as bacteria he observed, protozoans he observed, as well as he has been observed the yeast, he has been observed the red blood cells, that is RBC, red blood cells, etc. So many are there. That is the RBC are also called what? Red blood cells. So these he has been observed. Where he has been observed? He has been observed in a drop of water. Drop of water. So he has been observed all this invisible. The first person to observe the invisible microorganisms was Antony van Leuwenhoek. How he has been observed? With a specially developed lenses. They are called magnifying lenses, which was been specially invented by this Antony van Leuwenhoek, which is having the power of how much time? 300 times the magnification power. With the help of that magnifying lenses, he used it to observe that microorganisms. So in this way, he has been observed this about the microorganisms. Next is about Robert Hooke. About whom? Robert Hooke. Robert Hooke is also one more scientist who has been observed in the year of 1665. He has been discovered the cell. He has been discovered what? Cell. And where he discovered? He has been discovered the cell first in the bark of an oak tree. A bark of what? Oak tree. What in by bark actually means? The outer covering of a tree is called as a bark. The outermost layer of a stem is called as a bark. In Telugu we call it as beradu, outside part. And next in this oak plant he has been observed this cell. And how the cells are there means the cells are looking like empty spaces. They are looking like what? Looks like actually first it looks like a honeybee comb. How that honeybee comb will be? They are having the empty spaces. Empty spaces or empty boxes. They are looks like what? Empty spaces or what? Empty boxes. How they are there, Nana? Like this. So like this. This is one cell, this is one cell, this is one empty box, this is one empty box, this is one empty box. Each one box is treated as what, Nana? Cell. And here actually cell is a Latin language word. It is a Latin word. The meaning of cell in the Latin language is nothing but a little room. What is that, Nana? Little room. So this in this way he has been discovered the cell. He has been discovered about what? Cell. So now we are going to know about the activity 1. In that we are going to know about see the cell. See, as oak plant cannot be available at all the places, so that's why we have to be taking here the material mastic. You have to take a mastic and that has to be soaked in the water. That has to be soaked in the water for what? Half an hour. For half an hour. And from there you have to cut down into thin slices. You have to cut that into what? Thin slices. These thin slices you have to cut it out with the help of the knife or the blade, and one thin slice you take it out on a slide. You have to take it upon what? A slice. Sorry, take it upon what? And a slide, and a glass slide. And then you have to add some water. You have to add some water. Then you have to cover that glass slide with the help of cover slip. With the help of what? Cover slip. Then you can see with the help of microscope. You can able to see with the help of what? Microscope. Then you can see the cells present in the water mastic. Cells present in the what? Mastic. This is one activity 1. See, Nana, the cells present in the mastics are similar to the cells present in the oak plant. Why? Because both are dead cells. Both are what? Dead cells. So now let us take up another activity. In that activity we are going to see the live cells. That is living cells. That is activity 2. Activity 2. Activity 2. In the activity 2, what you have to take? You have to take an onion. You have to peel out that onion. You have to peel it. Okay. You have to remove the peel of that onion. That is outer covering of the onion. And then you cut a fleshy portion. A fleshy portion from onion you have to cut. You have to cut it. A fleshy portion has to be cut out. And then that fleshy portion you have to cut it out. And then that fleshy portion you have to break down into 2 equal parts. Break down into what? 2 equal parts. So that has to be break down into two equal halves. And then after breaking into two equal halves, between these equal halves, you have to see that a membrane is attaching, is connecting to the two equal halves. And that is nothing but a transculent. And a transculent membrane is connecting the both. And a transculent thin membrane is connecting the both two equal halves. From the thin membrane, you collect some piece of a thin membrane. That thin membrane, you collect it and then you keep that on the glass slide. You keep it upon what? On a glass slide. And same as usual, you add the water and you have to cover with a cover slip. You have to cover it with a cover slip. Then you can observe underwater microscope. You can able to observe it under microscope. Then you are able to see this onion peel cells or onion cells. How that onion cells will be? The onion cells will be like this. The onion peel cells will be like this. See, the cells are built like this. Next, activity 3. Activity 3. In that activity 3, we are going to know about the what nana? Cheek cells. You have to collect what nana? Cheek cells. How we can collect it? Means first you have to clean your mouth with help of water. You take the water and gargle the water and gargle it and you can spit it out that water. Then you take a wooden spoon. That is nothing but an ice cream spoon. That is called as spatula. That is called what? Spatula, or we can say it as a wooden spoon. You know, Nana, while you are eating the ice cream, you will get this wooden spoon. That wooden spoon, you have to scrape it gently. You have to scrape it what, Nana? Gently your inside cheeks. Okay, you can scrape your cheek gently with the help of this wooden spoon. Then that scraping you have to take it up on a slide, that is a glass slide. Then you can add some water, then you can add it with a cover it with help of cover slip, and then you can see under the microscope. You can see it under the water microscope. Then how the cells will be looks? The cells are looking like this. The cells are been looking like this. The cells has been looking like this. It is a nucleus. And these daughter like structures will be there. These daughter like structures are nothing but called what? Cytoplasm. What is that, Anna? Cytoplasm. Okay, cytoplasm. So this is what? This is nothing but cell membrane. It is nothing but what, Anna? Cell membrane. And the liquid like structure which is present here, this is called what? Cytoplasm. And whereas this round structure is called what? Nucleus. Is called what? Nucleus. Same here also, Nana. Here the daughter like structures which is present here, this is nothing but called what? That is cytoplasm. And here this is what? Nucleus. This is called what? Nucleus. And this is what? Cell membrane. That is called what? Cell membrane. And next here one more extra outer covering is present, Nana. This outer covering is called a cell wall. That is called what, Nana? Cell wall. So what you come to know that between the plant cell and animal cell, one extra structure is present in the plant cell, and a unique feature in the plant cell, that is called a cell wall, which is present in the plant cell, which is absent in the what, Nana? Animal cell. That is nothing but cheek cells. We are comes under the animal category. So onion comes under the plant category. So in the plant cell, the cell wall is present. Any plant cell you take, they have the cell wall. Whereas any animal cell you take, they won't have the cell wall. Only the cell membrane is present. That is the basic difference between the plant cell and what, Nana? Animal cell. So this first one is nothing but what, Nana? That is dead cells. Remaining two under comes under what? Living cells. So these are the three activities which reveals us about the nothing but which reveals about the cells. Okay friends, up to now we discussed about the cell and the activity which are concerned to the cell. Now we are going to see about the another important structure of the cell. What is that? That is nucleus. What is that, Anna? Nucleus. The important structure of cell is what? Nucleus. See, the nucleus in the year of 1831, Robert Brown has been discovered the nucleus. Discovered what? Nucleus. In the year of 1831, Robert Brown discovered the nucleus. But actually, it was in a belief that in the, that is Felice Fontana and some other scientists has been discovered the nucleus. First they observed the nucleus in the epithelial cells. In where, Nana? In the epithelial cells. What are these epithelial cells? Means that is outer layers of animals. The outer layer of animal is called what? Epithelial tissue or epithelial layer. In that cells, that is an outer layers, he has been observed the nucleus in the epithelial cells first. But he was not perfectly confirmed it is a nucleus or not. So that's why the credit of giving discovering of nucleus was given to what? Robert Brown. And actually where this Robert Brown discovered, you know, he has been observed in the epidermis. He observed in what? Epidermis of orchid plants. Of what, Nana? Orchid plants. So in orchid plants he has been observed this nucleus. And while he is observing how it is there, that is a nucleus. See here in the plant he has been observed this nucleus. For example, okay, this is a cells. Okay, in the cell centrally we have what, Nana? Nucleus. We have what? A nucleus. And while observing here, this is a nucleus, he said, and centrally located structure nucleus. And around this we have what, Anna? Cytoplasm. And moreover, see, cell was discovered in the previous video what we discussed. Cell was discovered by Robert Hooke in the year of 1665. But whereas the nucleus was discovered in what? 1831. That is nearly almost 150 years to 175 years gap is there between the discovery of the cell and discovery of the nucleus. And one more thing also Robert Brown observed. While he is observing the nucleus, he has been in the plant cell, he has been also observed that is stomatal cells. He also observed what, Anna? Stomatal cells. So he has been observed these stomatal cells also. These stomatal cells also useful for the exchange of gas actually in the leaves. Okay, he has been observed the stomata as well as the nucleus. So in that the discovery of nucleus has been came into existence. And later if you want to see the nucleus, then we have to do that is activity 4. Activity 4. In the previous video we come to know, Nana, what is that activity 3? That is how to observe the cell we did in the onion, that is in onion cells. Correct or not? Same procedure. You peel out that onion and cut the 2 onions in the, that is the 2 equal halves, and collect a thin membrane from this onion. You collect the thin membrane. The thin membrane should be spread upon the slide, that is a glass slide. Then you can observe under the microscope. How to observe? Here, Nana, after spreading upon this slide, you can add here nothing but water. At the same time, that is any stain, that is stain as well as the and cover slip, as well as what, Nana? Cover slip. So these you have to add. So you may get out, sir, what is by stain? Stain means a color giving substance. It may be methylene blue. If you add, that will be give blue color. Phenolphthalein you add, it gives orange color. Okay, and methyl orange also, that is the orange color. So like red ink, blue ink, any type of ink that is in the refills ink lane, that also you can add as a stain. The color giving substance is called what? Stain. And then you can observe the nucleus in the onion cells. So in the onion cells, you are able to observe the what? Nucleus. You are able to observe what? Nucleus in the center like this. And moreover, next if you want to know still clearly about this nucleus, we can go for activity 5. Go for what, Anna? Activity 5. In the activity 5, if you take it, this activity 5, if you take it means it will be in the cheek cells also you are able to observe that is in the cheek cells also you can observe the nucleus. For example, you will learn in the last video that is the cheek cells are like this, and these cheek cells will be like this, and these cheek cells in the center we have what, a nucleus like this. We have what, a nucleus like this, and this is a cytoplasm. This is what, a cytoplasm. And so the nucleus is present like this, and this is a cytoplasm will be present, and this is a nucleus. And same process, Nana, scrapping upon that cheek cells, in that you can add the stain. Same, you have to take the cheek cells, you have to spread it, that scrapping upon the slide, and you can add 1 or 2 drops of water, 1 or 2 drops of any stain, and then you can keep it with the cover slip, and then you can observe the nucleus in the cheek cells. You can observe it, what, Nana? Cheek cells. See, Nana, here in this activity, one thing you have to do. For example, this is a glass slide. On the glass slide, you are keeping this scrapping of the cheek. At that time, you are added 1 or 2 drops of water, 1 or 2 drops of stain like this. While you are adding with help of filter paper, and then you add it, and with help of water gently wash that slide, and you can extra stain should be drained out with help of filter paper. Extra drain should be drained means that has to be removed with help of filter paper. You keep beside that, extra stain will be observed. Then you can see the object very clearly. That is nucleus very clearly under that microscope. This is one of our activities. Now we have to know about that is a cell wall and next cell membrane. Cell wall and what, Anna? Cell membrane. You take here, Anna, cell wall is an extra layer. Okay, it is an extra layer present over the cell membrane. Present over what, Anna? Cell membrane. Whereas it is present only in the plant cell. In the previous video also I told. Whereas the cell membrane is a membrane surrounding the cell. Membrane surrounding what, Anna? Cell. Membrane surrounding the nothing but cytoplasm and cell organelles. Cytoplasm and what? Cell organelles. Cell organelles means the parts of the cell are called as a cell organelles. Okay, so that is the about cell wall as well as what, Nana? Cell membrane. And you may also get a cytoplasm. What by cytoplasm? See, Nana, cytoplasm means cyto means cell. Plasm means nothing but what? Liquid or fluid. So the liquid or a fluid present inside the cell. The liquid present inside the cell is called as a cytoplasm. Present inside the cell is called what? Cytoplasm. So these are the some few extra, that is, for example, this is a cell, this is a nucleus. Okay, Nana, and like this liquid is called what? Cytoplasm. I already that is in the last video I mentioned all the parts. And the like this, if you take a plant cell, an extra covering will be observed over that. An extra covering will be observed over that. That extra covering is nothing but called as what? Cell wall. That is called what, Anna? Cell wall. This extra covering is called what? Cell wall. So this is extra covering cell wall, and this is a cell membrane. This is cell wall. This is a cell membrane, and this is a nucleus. And this is what? Cytoplasm. So remaining organelles are present in this cytoplasm itself. So this is overally about the cell as well as the nucleus. Cell and what, Anna? Nucleus. Next we are going to know about the diversity of the cell. We are going to know about what, Anna? Diversity of the cell.
Thanks for watching. Please like, share and subscribe.